Welcome. My name is Associate Professor Charlene Kahler and I am the Deputy Director of the Marshall Centre. At the Marshall Centre we have many groups researching some of the most important issues facing the world today. Infectious disease does not affect all members of our community equally, with young children and the elderly more susceptible to adverse outcomes than healthy adults. Epidemiology relates an infectious agent to the incidence, distribution and possible control of the disease in our community. In the current pandemic of COVID-19, it has become clear that genome sequencing is a powerful tool to inform us of the evolution of variants and how this affects our vaccination strategies in different community groups. At the Marshall Centre, we undertake many epidemiological projects on infectious agents, examining their genetic variation. We are also developing novel technologies, including non-culture-based sequencing, to improve the rapid detection of infectious agents. The following statements will introduce each of our program leaders and their projects at the Marshall Centre.
Hi, I'm Dr. Alfred Tay, Research Fellow in Marshall Centre, Manager of Helicobacter Research Laboratory and the webmaster of helico.com. My research is all about Helicobacter pylori and its genetics. My research can be divided into two categories, that is, the clinical research and basic science research. In clinical research, I collect clinical isolates from the hospital and investigate the antibiotic resistance profile. This surveillance program monitors the antibiotic resistance in Western Australia and can be useful for doctors in dealing with difficult cases. In basic science research, I'm interested in finding out where did they come from, how do they become antibiotic resistant, and how do they spread among people. We can do all these by sequencing its genome. From the genetic information, we can use it to trace the origin of an infection, we can understand how they become resistant to antibiotics, and very interestingly, we can even use it to understand human migration. Now, Perth is a very interesting city because the population is established mainly by migrants all around the world. So my laboratory actually have a very good collection of Helicobacter pylori that represent every types of Helicobacter around the globe. We welcome scholars from around the world to collaborate with us to understand how these different Helicobacter pylori interact with each other. Recently, we paid a lot of attention in the Chinese Helicobacter pylori strains. Every year, about 500,000 of gastric cancer cases are reported from China alone. Treating Chinese Helicobacter pylori has always been shown difficult because of antibiotic resistance. Therefore, we are building collaboration partners in China to help us understand their local Helicobacter strains and combat antibiotic resistance. I always like to use Australia as an example. Because of the good work accomplished by all the doctors in Australia, the prevalence of Helicobacter pylori in Australia is only about 10 to 15%. Because of that, gastric cancer is already a very rare disease in Australia. Therefore, I believe that eradicating Helicobacter pylori will not only reduce gastric cancer rate, but also other gastric diseases, such as peptic ulcers and reflux symptoms.
My name is Dr. Daniel Knight, and I'm an Early Career Research Fellow here in the Marshall Centre. My area of expertise is microbial genomics and antimicrobial resistance, and my programme of research is centred on understanding the ecology and evolution of bacterial pathogens. My primary focus is the enteric pathogen Clostridium difficile, or C. diff. This bacterium is a leading cause of healthcare-associated infections worldwide and a major pathogen in many animal species. My One Health focused research has shed new light on the genetic and ecological factors which drive transmission of C. difficile between humans, animals and their shared environment. It proposes a novel zoonotic paradigm for C. difficile infection, with the environment acting as a conduit for the transmission of C. difficile as spores from animals to humans. The impact of this paradigm shift has informed guidelines and position papers from major health groups in Australia and internationally, with potential for improved diagnostics, surveillance and treatment of C. difficile infection across both clinical and veterinary medicine.
My name is Michael Wise. I'm an Associate Professor in Computer Science at UWA. I'm also a computational biologist and I'm an associate with the Marshall Centre. I do a range of things, but particularly I write programs, but I also use other people's programs. So an example of programs that I've written is a program called Phaser, which may or may not have anything to do with Star Wars. And what that does is look for phase variation in the genomes of microbial species. In other words, how they're able to switch their genes on or off. And I've been working with Charlene Kahler's lab for that. We're looking at why virulent species are virulent and what tools they use. Another thing I do is use other people's tools, in particular for evolutionary biology. In this case, I'm looking at coronavirus, but not just the current SARS, but the previous SARS virus and MERS virus and the 20% of common cold that is caused by yet another beta coronavirus. Basically looking at when they evolved, how they evolved. The other thing I've been done even longer has been to work with the Marshall Group on the evolutionary biology of Helicobacter pylori, in particular how it's followed us modern human migration out of Africa around 200,000 years ago. So you can track human migration by our own genomes or archaeology. You can also track it by the bugs that we carry, in particular this particular bug.
My research program examines the infectious agents Neisseria meningitidis and Neisseria gonorrhoeae. Neisseria meningitidis causes invasive meningococcal disease, a rare but rapidly fatal infection in the very young. My research program uses whole genome sequencing to examine the genetic variation of the vaccine antigens to ensure that the state's vaccination program is delivering maximum protection to the West Australian community. Neisseria gonorrhoeae causes the sexually transmitted infection gonorrhea, which is rapidly gaining resistance to most antibiotics and has resulted in a strain that is commonly untreatable with available antibiotics. My research program uses whole genome sequencing to identify novel antimicrobial resistance markers and to track the epidemiology of this infectious disease throughout the West Australian community. This information is used to inform intervention strategies to reduce community transmission. If you are interested in any of our projects, we will be delighted to hear from you.